Good evening and welcome to tonight's event. On behalf of the University of Southampton, I'd like to extend you all a very warm welcome to all of the staff, students, alumni and supporters who are joining us this evening. I'm Professor Mark Cragg. I work in the Centre of Cancer Immunology and one of the senior group leaders here. And tonight I'm delighted to be joined by two of my colleagues from the Centre, Dr. Tim Fenton, who will talk to us about some of his world-leading research and contributions to new research areas that he's been developing since we recruited him a few years ago. And also one of our postgraduate researchers, James Christian, who's going to talk to you about his current research project, what he hopes to achieve in that, and highlight what this project might mean for the future of immunology treatments. We're also joined this evening by Charlotte Moss, who's a clinical trial patient back in 2011. She's here to share her experience and to talk about her involvement with the Centre of Cancer Immunology. After their talks, we're going to have a Q&A session. And to submit your questions, please use the Q&A box, which you can see hopefully on the right of your screen. We'll hope to get us through as many questions as possible, so please feel free to enter them. And if we don't get to your question in time before the event closes, we'll still endeavour to respond to your questions. So without further ado, I'm now going to introduce Dr. Tim Fenton to present some of his research work. So Tim is an associate professor in cancer in Southampton. His research interests are focused on understanding cancer at the molecular level and how the disease tries to escape our immune system. As I mentioned, Tim was recruited to Southampton a few years ago, and he's going to introduce to you some of the innovative work that we're doing here in the Centre of Cancer Immunology. Over to you, Tim.
Thank you, Mark. Good evening, everyone. As Mark said, I'll talk to you a bit about some of the different work going on in the centre and how I think and why particularly I was attracted to come and work in Southampton and to bring my research here. So cancer, as I'm sure you'll all be aware, is a daunting, but we think a surmountable research challenge. I'm going to talk to you a bit tonight about where we excel in Southampton, in particular using the immune system to fight cancer, and how this has really represented a paradigm shift in the way we think about cancer and the way we think about treating cancer. And I'm going to talk about some of our own and some colleagues' work on understanding cancer as an ecosystem, what we call the tumour microenvironment, and how we can target various elements in the tumour microenvironment to kill cancer cells. And then I'm going to give a last slide on where some of the directions in which I think we're heading and where I'm particularly excited about progress over the coming years ahead. So the challenge, and this will come as no surprise to many of you, I'm sure, but it's a fact that 1 in 2 of us who were born in the UK since 1960 will go on to get cancer at some point in our lifetime. So clearly this is going to be a universal experience one way or another. But the good news is that since, if you think about the kind of modern era of cancer research, I guess, beginning in 1970, when the US government declared their war on cancer, at that point, 25% of us were surviving a cancer diagnosis for 10 years after that diagnosis. By 2010, thanks to cancer research, that proportion of people surviving long-term post-cancer diagnosis have risen to 1 in 2 of us. And Cancer Research UK, in their research strategy set out several years ago, are aiming to make that 3 in 4 of us surviving our cancer long-term within 20 years. So that's an ambitious target, but you can see how that progress really has been driven by cancer research and how we hope to improve that and eventually to almost all of us, or hopefully everyone, surviving their cancer long-term. So why was I attracted to Southampton? I moved my research here just over 2 years ago now, really because Southampton is a centre of excellence for cancer immunology. And cancer immunology has been a discipline for many, many years. And for a long time, it was an unfashionable discipline amongst cancer researchers and didn't receive as much attention or indeed as much research funding. But people, including many researchers in Southampton, worked a way to understand how our immune system responds to cancer for many years. And then just over really 10 to 15 years ago now, this really resulted in an explosion in our ability to treat cancer. This is a piece of data that continues to amaze me, really. This is a drug called ipilimumab, which is an example of a cancer immunotherapy. And what this drug does, it's an antibody, is it takes the brakes off our own immune response to cancer and allows our own immune system to kill cancer. And this just shows that the long-term survival of people with very advanced skin cancer, so metastatic melanoma. And it shows that although sadly, even with this drug, ipilimumab, at the time these trials were run, many of the patients would sadly still succumb to their cancer within the first 2 years. There was then a remarkable phenomenon occurring. Actually, the remaining 20% of patients, you can see this curve becomes a horizontal line at around the 2-year mark post-treatment. And at this point, people are surviving long-term with their very advanced skin cancer that would otherwise have killed them very quickly. So ipilimumab was clearly doing something very spectacular. And this led to an explosion really in the press, a lot of press attention, and I'm sure many of you will have read about cancer immunotherapy. So really, as I say, coming out of almost a backwater of research for many years, immunotherapy came centre stage into cancer treatment, with its success initially in advanced skin cancer, in some lymphomas, and also more recently in other cancer types, including lung cancer. And this resulted in the Nobel Prize being given for cancer immunotherapy several years ago now. So cancer immunotherapy is really the exciting place to be in cancer research in 2024. And in Southampton, we are pushing ahead with advances in cancer immunotherapy in the 3 main domains of cancer immunotherapy. So we have antibody therapy, and Mark, who introduced me, is a leading world expert in developing antibodies to target cancer and to activate parts of our immune system to target cancer. We're also working on strategies for improved vaccination against cancer, so therapeutic vaccines. Since COVID, we're all very aware of RNA vaccination and then the potential of RNA vaccination. That's applicable, we think, in a cancer context. And there are a lot of efforts underway to vaccinate against cancer in patients who have been diagnosed. And then checkpoint inhibition. And the drug I showed you, ipilimumab, the antibody, is an example of an immune checkpoint inhibitor. One of these drugs that can take the brakes off of our immune response to cancer and allow our T cells to attack cancer cells. And the important thing to note about immunotherapies is that compared to conventional chemotherapies and radiotherapy, which can have extremely nasty side effects, as I'm sure you're aware, oftentimes the cancer immunotherapies have fewer side effects that are generally milder. They're certainly not without their side effects, but they're generally milder and easier to manage. So just bear with me. How do we do good science in Southampton? Well, one of the things that we have here is a really strong connectivity with other parts of the institution. Particularly, we're based at the hospital site in Southampton General Hospital, and the Centre of Cancer Immunology is part of the wider School of Cancer Sciences spread across 3 buildings. But we're also very connected in with the clinical trials unit in Southampton, also with the biology departments, engineering over on the main campus, and with the other elements in the medical school, which really allows us to exploit our basic research discoveries and bring them into the clinic. I'm going to tell you a little bit about a couple of stories in which this has been done in Southampton in recent years, but I'm sure when I was showing you that previous piece of data that I was very excited about with ipilimumab, it won't have escaped your notice that actually I was talking about very good results for 20% of patients. But what about the other 80% of patients? Clearly, we have a long way to go, because what we want to be doing is for that line to be a horizontal line at 100% of patients surviving their cancer long term, not 20% of patients. And at the moment, there's a lot of work to be done in understanding what makes that 20% of patients gain a good response to ipilimumab? Why do those 20% of patients get essentially cured of their skin cancer while the other 80% don't derive this benefit? So that's really where a lot of our efforts, and particularly this is work in a number of the labs in the Centre for Cancer Immunology, including my own, is trying to understand if we can look at a patient at the time of diagnosis or in their tumour material and understand whether that patient is likely to gain benefit from our immunotherapy, or which immunotherapy they'll gain benefit from. The last thing we want to be doing is giving expensive treatments to people who will gain no benefit, and actually they will still potentially get side effects. So we want to spare people treatments that won't help them and give people treatments that will help them. So I briefly mentioned T cells. T cells are really a key weapon of the immune system against cancer. T cells are white blood cells. So they're involved, particularly effective at responding to viral infection, for example, but they also can attack and kill cancer cells in using very similar mechanisms as the way that they attack and kill virally infected cells. Now, I've talked already about ipilimumab. Ipilimumab is an example of a checkpoint inhibitor or a checkpoint blocker. And there are a number of antibodies shown here on the right, like ipilimumab, that have been approved for treatment of various cancers in humans. And what ipilimumab and these related antibodies on the right-hand side here do is they actually flick off switches that are on the T cell. So T cells are very potent weapons. They'll attack virally infected cells, they'll attack cancer cells, but they can also, if they're not regulated properly, they can attack our own healthy cells. And this is the cause of autoimmunity. So the immune system is a very powerful weapon and it needs to be very carefully controlled. So normally these immune checkpoints, things like CTLA4 and PD1 that I've shown on the right here, are off switches on T cells. And in cancer, the cancer can actually engage these off switches and shut down the T cell response to the tumour. What these immune checkpoint blocking antibodies do is they relieve this inhibition. So they stop cancer from flicking the off switch and they allow the T cell to remain active against the tumour. Over on the left, we have a different set of switches on the T cell, and these are switches that help to potentiate the activity of the T cell. And here I'm going to talk to you about a little bit of work that's been, or a large piece of work, led by a clinician scientist in the Centre of Cancer Immunology, Dr. or now Professor Sian Lim, who's a lymphoma doctor and researcher in the department. And she did a large body of work, starting with mouse models and progressing all the way through to a clinical trial called the REVA trial for patients with B-cell lymphoma, where she added an antibody that actually switched on a switch called CD27 on T cells to activate T cell responses against lymphoma. And this was combined with an existing antibody used to treat lymphoma against a molecule on the B cells themselves or on the lymphoma cells called CD20. And a lot of work on understanding the biology of that target, CD20, has been done within the Centre for Cancer Immunology in the department previously by Mark and others. So that's an example where the entire rationale for understanding why it would be useful to flick the CD27 switch on T cells in addition to targeting CD20 on the lymphoma cells was all worked out here in the department, in the Centre for Cancer Immunology, leading to this REVA trial for B-cell lymphoma. And now that's promoted yet another round of research in which Sian and colleagues in the CCI are developing even more effective CD27 antibodies, which act to engage that CD27 switch and turn T cells on to allow them to attack the lymphoma cells. One other piece of research I'd like to tell you about briefly this evening involves a collaboration is being led by an experimental pathologist in the department, Professor Gareth Thomas, And this is an example of a treatment for head and neck cancer. We've collaborated with Gareth and his team for several years, and these data on the left here are taken from a paper that we published with Gareth's group a couple of years before I came to Southampton. Now, this is a rather busy slide, but I'll talk you through the main elements of it. I mentioned earlier the cancer ecosystem, the tumour microenvironment. When a cancer is taken out of a patient, that's not just a ball of tumour cells, it's a ball of tumour cells, but intermixed with that are all kinds of other cell types from our own body that have been kind of co-opted into the tumour. So blood vessels, for example, immune cells, stromal cells. So there's a lot of different tissue in that tumour. And on the top left, those four pictures you can see at the top left one is that those brown dots there are actually T cells that are in this tumour. So that you can see in parts of the tumour, they get full of T cells. And that's why if we can take the brakes off of the T cell responses, sometimes we can get a very strong anti-tumour response. These are T cells that have gone in to attack the tumour, but the tumour is suppressing their activity. Other tumours, like the one immediately below the picture with all the brown dots, has very few T cells in it, but what it has lots of, and we found this with Gareth, is that it has lots of a protein called smooth muscle actin, that SMA, which is the brown stain there in the bottom right picture. And that is staining, essentially scarring of fibrous tissue that's formed in the tumour. So what Gareth's lab showed through a series of very elegant experiments in cell and mouse models is that if you have tumours that have a lot of this scarring in the tumour, that builds up a network of what we call collagen that keeps the T cells out. So if you have a lot of scar tissue in the tumour, you can't get the T cells into the tumour. So with this in mind, Gareth and his team devised an alternative way in which to treat tumours. And they showed, as I say, using animal models, that actually, if you can target the scarring process, so the cells that mediate this are called fibroblasts. If you can target the fibroblasts, so we're not trying to target the tumour cells directly now, we're targeting the fibroblasts in the tumour, you can actually melt that scarring away and allow the T cells into the tumour. Then when you use your immune checkpoint blocking antibody to activate the T cells, you get a very effective anti-tumour response. So you don't just need to switch the T cells on sometimes, you actually need to get the T cells where they need to be in the tumour and then switch them on. And so this is really exciting work. And this has led to a clinical trial in head and neck cancer. And this is a phase 2 clinical trial. So the safety of the drug being used is already known. So we've gone straight to trying to show how effective it is. So I just very quickly, because I'm running short of time, just take you through a couple of areas in which I think things are going to be progressing in the coming years. Getting a really deep understanding of how our antibodies work at the atomic level resolution, improving the prediction of the peptides that we use to design better vaccines. These are all things happening in the CCI. Generating better cell and animal models for understanding the interaction between cancer cells and our immune cells. Improved DNA sequencing to understand how tumour cells differ from our own immune cells so we can target them better. And understanding, as I say, the tumour ecosystem. So really building up that picture of all the different cell types in the tumour and how we can modulate their behaviour to get better therapy. And this is all relying on collaboration between ourselves, but also increasingly across the disciplines and with industry partners, and also trying to look into, of course, preventing cancers. And there's work going on in the CCI on that front as well, particularly with a major trial in prevention of lung cancer. So I thank you for your attention, to answer questions later and leave it there. Thank you.
Fantastic. Thanks very much, Tim. So hopefully Tim's given you a bit of insight into the type of work that we're doing in the centre, about how many people are involved in each study, and the fact that we try to bring together clinicians with basic scientists and scientists across multiple different sites, both at the university, nationally and internationally. So it's a complex area, and it involves a lot of people to try and make good progress.